Welcome back to the deep dive. Today, we are tunneling into the immune system's most essential and maybe most misunderstood control center, the lymph node.
Yeah, we often hear the lymph node described as this quiet, organized war room. It's where T cells and B cells meet up, process information,
Right.
and design that highly specific, long-lasting plan we call adaptive immunity.
The strategic planning headquarters.
Yeah.
But that's really only half the story, isn't it?
Oh, absolutely.
When a serious infection hits, say a bacterial threat out in the periphery, those pathogens drain right into that LN. And if they pass through, you're looking at a systemic infection, maybe sepsis. So the LN has this incredibly demanding dual identity.
Precisely. It must simultaneously be the rapid response guard dog delivering immediate kind of brute force innate defense to stop pathogens from spreading.
And at the same time.
And at the same time, it has to host that quiet strategic planning session for long-term T-cell memory. These two jobs, rapid chaos versus organized precision, they're fundamentally at odds.
And the sources we've analyzed today reveal the amazing cellular mechanism that handles this impossible conflict. The dendritic cell, or DC.
Right.
Our mission today is to understand how DCs serve as the immune system's true rheostat, dynamically tuning the LN to prioritize immediate containment, even if that means, temporarily compromising the perfect organization it needs for long-term immunity.
It's the central biological puzzle. How does an organ manage this large-scale chaotic inflammation that's necessary for rapid containment without completely undermining the structural integrity and the precision needed to generate a robust T-cell memory response later on?
And what's so fascinating is that the sources show DCs don't just manage the conflict.
No, they intentionally cause the chaos first.
Okay, let's unpack this immediate response then.
Yeah.
What happens the moment a peripheral infection like that bacterial threat first hits the draining LN?
Well, the initial phase is pure damage control. You have macrophages, the immune systems, garbage collectors, and filters, right?
Right.
They're strategically positioned along the lymphatic sinuses to immediately intercept any incoming microbes. This is followed by a rapid activation and accumulation of existing natural killer cells, or NK cells, already waiting in the area.
But if the threat is big, you need serious, fast reinforcements. And those have to come from the bloodstream.
Absolutely. The heaviest hitters are the circulating immune cells, neutrophils and monocytes, NEUTs and MOs.
Yeah.
They are rushed into the LN via these specialized entry points called high endothelial venules, or HEVs.
And historically, we thought this created a kind of firewall, right? Confined to the edges of the LN.
Exactly. Immunologists believe this recruitment created a rigid, innate cell firewall, mostly at the outer boundaries. The inner T-zone, that core area where T cells live, was thought to remain relatively pristine, reserved for DCs to meet T cells in peace.
It sounds like a beautifully compartmentalized fortress. You have the innate cells fighting on the perimeter and the DCs handling intelligence and strategy in the inner sanctum.
And that is the first major revelation from this new data. That strict, clean compartmentalization is a myth during an active infection.
Wow.
That belief just fundamentally overlooked the speed and the severity of the innate response. The innate cells, the NEUTs and MOs, they do not stay confined to the periphery. They rapidly and robustly infiltrate the deep LN parenchyma.
So they go right into the core.
Right into the core. The organized inner T-zone, the very place where adaptive responses are supposed to start, becomes instantly flooded and converted into an innate battleground.
That is a massive counterintuitive shift. If we thought the T-zone was pristine, how did the researchers prove that this influx of innate cells is happening so fast and so selectively?
They used a brilliant technique called serial intravascular staining, or SIVS.
Okay, SIVS. Let's break that down for a second.
Right, so you can think of it like tracking mail with colored stamps based on arrival time. By injecting different fluorescent antibodies intravenously at timed intervals, say a green label for cells entering in the first hour, then a red label for cells entering in the next hour, they could track the kinetics of infiltration.
So they measure the traffic jam in real time.
Exactly. And by comparing these stamp cells, they confirmed that the influx rates were selectively and rapidly increased for neutrophils and monocytes compared to every other lymphocyte. This shows a preferential urgent bias towards pulling these specific innate cells from the blood directly into the LN core.
So the LN is being flooded with what are essentially chaos agents, and they're being pulled right into the strategic center. Something has to be directing this traffic. That doesn't happen by accident.
And that is the core of the DC rheostat function. The conventional dendritic cells are responsible for this massive polarized infiltration.
Ah,
They are the central sensory cells that sense the pathogen and immediately modify the entire LN architecture to establish what the sources call the pro-innate state.
so they're not just passive antigen presenters.
Not at all. They are actively dictating where the innate immune system mobilizes inside the LN.
That's amazing. The cell known for initiating long-term precision is actually the conductor of short-term chaos.
So we know DCs are the conductors driving this rapid selective influx from the blood into the T-zone. But how do they actually achieve this huge cellular mobilization?
Yeah, how do they do it?
It's a sophisticated 1-2 punch of molecular signals. Think of it as providing both the GPS coordinates and the physical adhesion highway.
Okay, let's look at the GPS first. How do they signal the general location?
So mechanism 1, chemokine gradients. DCs act as the primary source for the chemotactic signals, the chemical attractants. After sensing the infection, resident DCs rapidly upregulate expression of a wide array of inflammatory chemokines, and they create this polarized chemical gradient, a breadcrumb trail, that extends throughout the LN parenchyma, pointing directly toward the T-zone.
What specific chemicals are we talking about here?
Signals like CCL5, CXCL1, CXCL2, CXCL9, and CXCL10. These are the molecules that neutrophils, monocytes, and NK cells are basically hardwired to follow.
And they confirmed that the DC was the source.
They did, using a really clever genetic tool, the ZBTB46 DTR chimera model.
Wait, okay, let's pause on that jargon for a moment. For you listening, what does using a chimera model like that actually let researchers do?
Right, so a chimera is essentially a mouse rebuilt with specific components. The ZBTB46 DTR chimera allows researchers to specifically and temporarily wipe out DCs from the system.
Like a delete button for DCs.
Exactly. And when you deplete the DCs using this model, you see a massive, significant loss of expression for most of those inflammatory chemokines throughout the LN. That proves conclusively that the DC is the primary source, creating that chemical GPS signal.
So the DCs are the sirens screaming, "Come here!" But if you need hundreds of thousands of cells to enter a densely packed organ quickly, chemical signals alone, that might not be fast enough. You'd need physical assistance.
Precisely. And that brings us to mechanism 2. ICAM1 and the haptotactic matrix, the adhesion scaffold.
The highway.
The highway. DCs rapidly upregulate intercellular adhesion molecule 1, or ICAM1, right after immunization. ICAM1 is an integrin ligand, which is basically a sticky protein that immune cells use to tether themselves as they move.
So the DCs don't just stand there shouting directions. They start building a literal road for the other cells.
That's the perfect analogy. Visualization confirmed that DCs quickly form what the researchers called an adhesion molecule-rich matrix. This is a physical structure that extends seamlessly from the HEVs, those blood vessel entry points deep into the paracortex. This physical highway facilitates both efficient entry and the subsequent high-speed migration of innate cells into the deep T-zone.
How did they prove that the ICAM1 on the DC itself and not on, say, some other cell was the key?
Another specific genetic tool, the DC-ICAM1 Tech-O chimera.
So a knockout.
A very specific knockout. They essentially took the ICAM1 adhesion molecules away, only from the DCs. And what happened? It significantly reduced the total number of neutrophils and monocytes recruited.
Wow.
And crucially, the cells that did manage to enter stayed closer to the HEVs, the periphery, and failed to penetrate the deep T-zone efficiently. The highway was closed, and traffic just couldn't move deep into the organ.
That is surgical proof. Now, do all types of DCs participate in this massive recruitment equally?
Yeah.
Or is there a specialist?
No, and this is a critical nuance. This rapid, large-scale recruitment is predominantly dominated by resident DC2s.
Okay, the DC2s.
They looked at mice deficient in IRF4, which impairs the function of these specific DC2s. In these mice, the innate cells, the NUTs, MOs, NKs, they were severely misdirected. They just stayed near the HEVs and failed to infiltrate the T-zone.
So the DC2s are the main architects of this.
They are the dominant primary architects and drivers for innate cell influx into the core of the LN.
So to recap this critical phase, DCs act as the LN central command. They move toward the T-zone while laying down both guidance cues, the chemokines, and a physical adhesion highway, the IPM1. This is a massive, polarized effort, driven primarily by resident DC2s. So the question we need to address now is, this DC-driven chaos is essential for immediate survival, but what is the actual biological cost of turning the strategic T-zone into an innate battleground.
Right, and the necessity of this aggressive early action is undisputed. When you're facing serious threats like P. aeruginosa infection, the DCs organize the arriving neutrophils to form this dense barrier or cellular aggregate.
Quarantine zone.
It effectively quarantines the bacteria, preventing systemic dissemination. You can measure this by the drastically reduced bacterial load in the spleen.
So DC involvement literally saves the organism from sepsis. That's a huge win.
It is. But if you remove the DCs, the neutrophils become dispersed, they fail to swarm properly, and pathogen control just falls apart. You see an increased bacterial burden everywhere.
So they're essential for containment.
Yeah.
But survival comes with severe side effects. The architectural disruption.
Yes.
Tell us about the damage. How does the structure of the LN actually suffer?
The extensive influx of innate cells, particularly the sheer number of neutrophils, severely disrupts the carefully structured LN architecture. Microscopically, the T-zone, which should be densely packed with T cells waiting for antigen, it appears sparse and physically less dense.
So it's literally being torn apart.
B-cell follicles are also frequently disrupted, and even the HEVs, the entry points, show signs of damage indicated by reduced structural markers.
So the immediate defense is tearing up the infrastructure required for the long-term defense.
And it's not just physical disruption. DC-mediated activity promotes the expression of inducible nitric oxide synthase, or INS, across the tissue.
INS, that's a powerful killer molecule.
It's fantastic for killing bacteria. It's a potent antimicrobial. But here's the biological dilemma.
Yeah.
It is also a powerful suppressor of both T and B cell responses. The very molecules DCs promote for immediate defense create this toxic chemical environment that suppresses the initiation of the long-term adaptive response.
That is the ultimate trade-off, a true biological sacrifice. You suppress the quality of your T-cell response now to survive the infection today.
Exactly.
What was the physical consequence of all this chaos for the adaptive T cells, the ones that are supposed to be getting activated?
The physical displacement was dramatic. Researchers tracked antigen-specific T cells, the very cells that need to meet the DC to get primed, and found they were almost completely excluded from the neutrophil-rich, inflamed regions of the T-zone.
So they were locked out.
They were forced to the sidelines, relegated to the less inflamed margins of the LN, where antigen presentation just wouldn't be as efficient. It was like forcing the general to plan the war from the cafeteria instead of the war room.
But how did they know it wasn't just the chemical aftermath, the INOS, but the neutrophils themselves that were causing the suppression?
That's a crucial question. They performed an elegant experiment. They depleted the neutrophils using a targeted antibody.
So they just took them out of the equation.
Just took them out. And when they removed the physical presence of the neutrophils, the T cell distribution was immediately restored across the tissue.
Wow.
And critically, early T cell activation significantly increased. They measured this by finding more activated T cells and a greater abundance of antigen-specific CD8 T cells.
So that confirms it.
It definitively confirms that the extensive DC-driven innate cell influx actively limits the magnitude of early adaptive responses due to both physical and chemical interference.
But the critical thing to remember here is that this disruption is not permanent. The system has to be designed for recovery.
Exactly. The DCs, having caused the initial chaos necessary for containment, must then initiate the cleanup. They have to transition the LN from that pro-innate state back to the pro-adaptive state.
It's a transition from general and architect of chaos to the head janitor.
Yeah.
How quickly does this transition happen, and what's involved in the cleanup?
It's surprisingly quick. Neutrophils are very short-lived cells, and they naturally undergo apoptosis, or programmed cell death. So you see a natural decline in their numbers after about 24 to 36 hours.
And who makes up the cleanup crew?
Well, it's composed of the DCs themselves and the monocytes they originally recruited.
So they clear their own mess.
They clear their own mess. They clear the dying neutrophils via phagocytosis, a specialized cleanup process called efferocytosis. This was directly observed. They found molecular debris from neutrophils, signals like MPO and Li6G, inside the phagocytic DCs and monocytes.
And that's vital.
It's absolutely vital because it removes the source of inflammation and structural obstruction, allowing the LN architecture to regain the density and organization required for potent adaptive responses.
So the battlefield is cleaned and reconstruction can begin. But does that initial innate presence leave a lasting structure? Does it affect the quality of the future adaptive response?
It does. While the neutrophils are efficiently cleared, the polarized distribution of monocytes remains for a longer period. This creates distinct functionally different microenvironments across the tissue. You have monocyte-rich zones versus DC-rich zones. And this spatial polarization is not just passive geography, it actively drives heterogeneity in T-cell differentiation.
Meaning different types of T cells are generated in different neighborhoods.
Precisely. The effector T cells, the cells that will eventually leave the LN to fight the infection, are generated primarily in those monocyte-rich zones. This sustained spatial arrangement, a legacy of the initial innate influx, ensures that the quality and functional specialization of the long-term adaptive response are optimized.
So it provides both immediate containment and better memory down the line.
That's it.
So final synthesis. DCs are dynamically adaptable rheostats. They coordinate the dual functions of the LN by first prioritizing immediate defense at the clear expense of early adaptive efficiency, and then diligently resolving that damage to enable maximal specialized adaptive responses later.
This capability fundamentally redefines their role. I mean, they are not just presenting antigen. They are actively and temporally organizing the entire cellular landscape of the immune response, from the moment of threat containment to the generation of specialized memory.
So what does this all mean for us? The classic image of the lymph node as a pristine, compartmentalized fortress is officially inaccurate.
Dead in the water.
It's a dynamic battlefield where organization is temporarily and strategically sacrificed for survival, a trade-off entirely managed and synchronized by the DC rheostat.
And the implications are vast, especially for vaccine design. These findings highlight a critical temporal synchronization problem.
How so?
Well, most current adjuvants, the ingredients used to boost vaccines, they just focus on activating DCs and achieving massive innate cell recruitment. But these sources suggest that excessive or prolonged inflammation, the very chaos DCs promote early on, could actually be detrimental to generating the strongest possible adaptive memory response later.
So we need synchronization, not just massive activation.
Exactly. Precision synchronization.
It makes you rethink what constitutes a good adjuvant. If excessive early innate chaos limits adaptive immunity, how might future vaccine adjuvants be designed to retain the local containment benefit of innate cells, like rapid life-saving neutrophil aggregation, without triggering the subsequent large-scale architectural disruption that hinders optimal T-cell priming? That balance between immediate survival and future strategy. That's the ultimate biological engineering challenge, something for you to mull over.